the great mystery of the origin of life. How did it originate? Maybe it all happened the way it's shown in science fiction movies? Once, gray-blue giants flew to the planet and seeded the Earth with the necessary molecules. Well, nature and time brought this process to its logical conclusion. This scenario doesn't seem all that fantastic compared to, for example, the achievements of modern biotechnologists. Meet Xenobot, a creepy, programmable organism made from living cells. You could say it's a biological robot. Under the microscope, you can see small clumps moving chaotically in the liquid environment of a petri dish. They move around, self-organize, and even carry loads. But if you flip the clump over, it will just stay there, like a turtle turned on its back. Their behavior is reminiscent of microscopic flatworms, or even tiny tardigrades, which have a complex body structure despite their small size. But the similarity in behavior is not deceptive, because the clumps are made up of only two types of cells: frog skin cells and frog heart cells. First, they extracted stem cells from frog embryos and differentiated them into heart cells, which have the ability to contract, and skin cells, which do not have this property. Then they combined these active and passive components, using the natural tendency of the cells to stick to each other. When encountering individual cells along their path, the clusters gather them into small piles. And if you cut the Xenobot in half, it will rejoin itself, just like the T-1000 from the movie Terminator 2. But maybe it all happened without any alien technologies, without a landing party of little gray men, and life was delivered to the third planet from the Sun by some other means? After all, the theory that life on Earth originated from space is being actively pursued by many scientists. For example, a reputable international scientific team led by Australian immunologist Edward Steele, with the participation of astrobiologist Chandra Wickramasinghe, honorary professor at Buckingham University and director of the Center for Astrobiology, published a review article in the journal Progress in Biophysics and Molecular Biology, where they presented an analysis of the hypothesis of the extraterrestrial origin of life on Earth. Since the 1980s, Wickramasinghe, a British physicist, astronomer, and astrobiologist, has noted traces of organic matter on comets, clear signs of microbial life. The scientist believes that many genetic features could not have arisen through random mutations, and allows for the possibility that comets that reached Earth contained frozen biomaterials. Proof of their viability came from an experiment by Swedish scientists studying tardigrades in outer space. The samples were divided into three groups. One of them, upon arrival in orbit, was exposed to vacuum conditions and subjected to cosmic radiation. Another group was exposed to ultraviolet A and B radiation from 280 to 400 nanometers. The third group of animals was exposed to the full spectrum of ultraviolet from 116 to 400 nanometers. All tardigrades were in a dormant state. After 10 days spent in outer space, almost all the organisms were desiccated. But on board the spacecraft, the tardigrades returned to their normal state. Most of the animals exposed to ultraviolet radiation with a wavelength of 280 to 400 nanometers survived and were able to reproduce. Only 12 animals from the third group survived. Nevertheless, the survivors were able to produce normal offspring, although their fertility was lower than that of the control group that remained on Earth. This experiment proved that tardigrades can survive in space conditions. At the same time, the journal Geochemica et Cosmochemica Acta published the results of a study of dust particle samples collected by NASA's Stardust spacecraft from the surface of Comet Wild 2. In these samples, scientists from the University of Arizona found minerals that could only have formed in the presence of liquid water. Here's another fact. Spectral analysis of Halley's Comet revealed the presence of organic molecules in it. If you add to this the already proven fact that comets can contain liquid water, it turns out that travel becomes quite easy for microorganisms. Especially since, as we can see from tardigrades, some don't even need water to survive cosmic temperatures and radiation. These tiny animals, returning in the Cambrian, pose a serious challenge to traditional thinking. Their space-resistant qualities are fully consistent with evolution in extraterrestrial conditions and are incompatible with purely terrestrial conditions. both modern and those from 500 million years ago. There is also interesting data on a question that became one of humanity's very first memes. Is there life on Mars? Almost half a century ago, 45 years ago, two Viking landers became the first American spacecraft from Earth to land on Mars. They took the first high-quality images of the planet, studied the planet's geographical features, and analyzed the geological composition of the atmosphere and surface. But the most interesting thing is that they also conducted experiments searching for microbial life in the Martian soil. Overall, these experiments to detect life produced unexpected and controversial results. One of the experiments, the labeled release experiment, showed that Martian soil gives a positive result for metabolism. But no traces of organic material were found. Where could metabolism come from if there was no organic matter? The protocol for this experiment was developed by Gilbert Levin. During the experiment, both landers, Viking 1 and Viking 2, collected samples of Martian soil, added a drop of diluted nutrient solution to them, and then observed the air above the soil, waiting to see signs of metabolic byproducts. Since the nutrients were labeled with radioactive carbon-14, if microorganisms in the soil metabolized the nutrients, they would have produced radioactive byproducts, such as radioactive carbon dioxide or methane. Before the launch, the experimental protocol was tested on a wide variety of Earth soils in places inhospitable to life, from Death Valley to Antarctica. In every case, the experiments gave a positive result, detecting life. Then, as a control, scientists heated the samples to 160 degrees Celsius to kill all forms of life. And, as expected, they got a negative result. To confirm that the experimental procedure would not give a false positive result, scientists tested it on sterile soil, which also gave a negative result. For decades, Gilbert Levin was not allowed to publish a scientific paper revisiting the results of that old experiment. That experiment was considered a failure, and only relatively recently was he able to publish about it. And not only because the scientific publication by Gilbert Levin and his colleague Patricia Stratt was, according to the authors, so thoroughly polished that it was impossible to find fault with it. The point is also that in 2014, the Curiosity rover's laboratory detected organic molecules on Mars for the first time. The SAM laboratory detected, among other things, methane, chlorinated hydrocarbons, and other organic molecules there. Another significant discovery was made by astrobiologists from Cardiff University. They analyzed fragments of a meteorite that fell in Sri Lanka on December 29, 2012. Chemical analysis showed a relatively low density of the material, as well as a high content of carbon and organic substances. Using electron microscopy, scientists were able to discover round structures inside the meteorite fragments with a diameter of about 0.1
мм,
resembling fossilized algae. On the surface of the structures, threads could be seen, which may be the flagella of these algae. As an argument in favor of the extraterrestrial origin of the structures, rather than contamination on Earth, scientists point to their location inside the meteorite fragments and their low nitrogen content. And in 2018, a report was published by Russian specialists with the results of a study of microbes from cosmic dust that had stuck to the outside of the ISS window. Viable bacteria were discovered, and to explain how they ended up at an altitude of 400 kilometers above the Earth, two versions were considered: a hypothetical ionospheric elevator mechanism from Earth, and, as an alternative origin for these microbes, space. There is another side to the process, because not only tardigrades and algae can be space travelers. Scientists are seriously considering viruses as carriers of life through space. For example, there is a study suggesting that the so-called Cambrian explosion, a sharp increase in the biodiversity of skeletal fauna about 540 million years ago, was caused by the arrival of viral genes on Earth. In the fossil remains from that time, representatives of all animal types that exist today, including chordates, appeared within a short period. An analysis of the coevolution of retroviruses and their amphibian and fish hosts showed that viruses appeared in the ocean 460 to 550 million years ago at the beginning of the Paleozoic era. According to astronomers' calculations, on the eve of the Cambrian explosion at the end of the Ediacaran period, Earth was subjected to meteorite and comet bombardments associated with the solar system passing through a giant molecular cloud. Such events are not uncommon by cosmic standards, since as the Sun orbits the center of the galaxy, it passes through many giant molecular clouds. This is a well-established scientific term. Just imagine an entire galaxy, or perhaps even the whole group of galaxies that includes ours, could make up a single connected biosphere, not just a cosmic sector of space. The result of this, on one hand, is mass extinctions, and on the other hand, the active replenishment of Earth's gene pool with cosmic genes, which are integrated into terrestrial genomes and drive evolution forward. And viruses are the perfect candidates for such integration. They make up at least 8% of the human genome, which allows us to conclude that natural selection in humans and their ancestors took place in partnership with hundreds of viruses. Their resilience makes them virtually invulnerable. In the course of evolution, some viral genes that became fixed in the genomes of cellular life forms began to perform important functions for the organism. For example, the placenta, this red circle on the right, allows both humans and this mouse to begin life inside the mother's womb. It's possible that this remarkable organ would never have appeared if our distant ancestors hadn't picked up a very strange infection somewhere. The PEG10 gene, which is necessary for placenta development, apparently was borrowed by ancient mammals from a mobile genetic element called a retrotransposon. A retrotransposon is something like a simplified virus that has almost lost its infectiousness, that is, its ability to transfer from one host to another's genome. But it can still reproduce inside host cells and insert its copies into the host's genome. They are usually passed on only by inheritance, from parents to children, but sometimes, although extremely rarely, they can still be contracted like viruses. This is not the only case of this kind. The enzyme telomerase, which is responsible for restoring the ends of chromosomes that shorten after each replication, traces its ancestry back to a retrotransposon. Here's another example. One of the genes that plays a key role in learning and memory is the ARC gene. This virus-like gene is essential for the long-term storage of information in the brain. The protein behaves like a virus and serves as a platform for neuron interaction. ARC kind of opens a window through which memories solidify. And this window can't open without this gene. A group of scientists led by Jason Shepard, an associate professor of neurobiology at the University of Utah, who has devoted more than 15 years to studying proteins, believes that a retrotransposon, the ancestor of a retrovirus, entered the mammalian body millions of years ago, which later led to the development of the ARC protein. A mouse with the ARC gene turned off is unable to learn or form long-term memory. After finding cheese in a maze, it forgets the way to it the very next day. But that's not all. Viruses are not only very good at integrating into the genome, but also at cooperating, disguising themselves, and exchanging messages with each other. This discovery was made by scientists from the Weizmann Institute, led by genetic microbiologist Rotem Sorek, who published the research results in the journal Nature. The authors of the study suggested that one of the proteins serves as a means of communication between viruses, altering their behavior. After 2.5 years of experiments on Bacillus subtilis bacteria, they identified this substance. It turned out to be a viral protein released from bacteria killed by bacteriophages. By the way, if you look at what bacteriophages look like and what they do, that is, viruses that infect bacterial cells, the question of the extraterrestrial origin of these monstrous mechanisms hardly even arises. So, the communication protein between bacteriophages is a viral peptide made up of 6 amino acids, and it is produced when a phage infects a bacterium and signals to other phages that the host is infected. When the number of signaling peptides, and therefore the number of infected cells, reaches a critical level, all the viruses, as if on command, stop active reproduction and go dormant. If it weren't for this deceptive maneuver, the bacteria could organize a collective defense or be completely destroyed, depriving the viruses of the opportunity to continue parasitizing them. Viruses lull their victims into a false sense of security and give them time to recover. The peptide that helped them do this was called arbitrium. Further research showed that viruses are capable of making even more complex decisions. They can sacrifice themselves during an attack on the cell's immune defenses to ensure the success of the second or third wave of assault. They are able to move in a coordinated way from cell to cell in transport vesicles, exchange genetic material, help each other mask themselves from the immune system, and cooperate with other strains to take advantage of their evolutionary benefits. There is a high probability that even these amazing examples are just the tip of the iceberg. And a new field has even emerged to study the social life of viruses. Viruses. It's not that viruses have consciousness, but social connections, a language of communication, collective decisions, coordination of actions, mutual assistance, and planning can quite reasonably be considered signs of intelligent life. Here are the most pressing examples. Under the shell of the famous ball with spikes is a short RNA molecule, a chain with 29,903 nucleotides. For comparison, our DNA contains more than 3 billion of these nucleotides. It's quite a simple structure by comparison, but the virus doesn't need to be complex. The main thing is to become a key component of a complex system. Viruses can be compared to Somali pirates who capture a huge tanker with a tiny boat. They don't need the entire control algorithm of the captured cell. A short code is enough to make the entire operating system of the cell work for it. For this task, its code is perfectly optimized. Inside the cell, the virus comes to life only to the extent that the system's resources allow. In a simple system, it is capable of dividing and controlling metabolic processes. In a complex one, such as the human body, it can also use additional options, for example, reaching a level of information processing that borders on intelligent life. But why do viruses need this at all? Sacrificing themselves? Helping each other? Improving the process of communication? What is their purpose if they are not living beings? Strangely enough, the answer has a direct connection to us. Basically, a virus is a gene. The primary task of any gene is to copy itself as much as possible in order to spread through space and time. But in this sense, a virus is not much different from our own genes, which are also primarily concerned with preserving and replicating the information encoded in them. In fact, the similarity is even greater. We ourselves are a bit like viruses. The human body has accumulated about 98,000 retroviral elements that our ancestors were once infected with. Now they make up 30 to 50 families, which are divided into almost 200 groups and subgroups. According to geneticists, the last retrovirus that managed to become part of our DNA infected the human population about 100 to 150,000 years ago. Our ancestors experienced a pandemic then. What are relic viruses doing now? Some of them don't manifest at all. We actually don't know about these manifestations. Others work quite successfully, as we've seen. They protect the human embryo from infections, restore chromosome replication, and are involved in thinking and memory. But maybe the mission of viruses is much more significant. It was once believed that bacteria were only harmful. But as knowledge accumulated, it turned out that they are not only beneficial, but in many cases, absolutely essential for life. In the same way, perhaps soon people will stop demonizing viruses. Often, they bring us disease and death. But what if their purpose is not the destruction of life, but directed evolution? After all, we influence each other's evolution not just as environmental factors. Our cells are directly involved in the assembly and modification of viral RNA. And viruses come into direct contact with the genes of their hosts, inserting their genetic code into their bodies. It turns out that a virus is one of the ways our genes communicate with the outside world. However, it's hard to shake the thought that viruses, among other things, can arrive not only riding on comets, but also in a dish like this. After all, you can bring not only bacteria, viruses, and organic molecules. You can even bring almost fully formed organisms. For example, there is a huge amount of evidence pointing to the extraterrestrial origin of octopuses, squids, and other advanced cephalopod mollusks. An enlarged brain, an intricate nervous system, complex camera-type eyes, extraordinary fluid bodies that instantly change shape and color. All these facts are very difficult to explain with standard gradual evolution. Take, for example, the fact that octopuses actively edit their RNA, while their DNA remains highly conserved. In the vast majority of animals, spontaneous mutations in DNA determine their further development, while squids and octopuses are able to purposefully change their RNA to better adapt to their surrounding environment. Such modifications affect protein production, allowing the organism to better adapt to existing conditions without resorting to actual mutations in the genome. Back in 2015, scientists discovered that a squid can edit up to 60 RNAs in its nervous system as it tries to better adapt to changing temperatures. And recently, an analysis of thousands of RNA samples showed that similar abilities are also present in octopuses and cuttlefish, mollusks that mainly inhabit warm seas near the coast. But let's take a step back and ask the main question. Even if the unified biosphere is not just our small planet on the outskirts of the Milky Way, but an entire group of galaxies with billions upon billions of living planets, then where is the origin? Where is that star system where it all began? And how exactly did everything come together so successfully there that, in the end, life began to spread on a cosmic scale? After all, at the moment, there are several main scientific theories about the origin of life, and about twice as many non-scientific ones. These include the primordial soup theory and the clay, rather than water, origin theory, where the optimal environment for the accumulation and increasing complexity of organic molecules is found in clay minerals. There's also the theory that life originated at the bottom of the ocean near black smokers, where the harmful radiation of the young sun couldn't reach. There are also protocells, the RNA world hypothesis, and even the remarkable endosymbiosis theory. Back in the late 19th century, it was noticed that chloroplasts, the organelles in plant cells responsible for photosynthesis, replicate independently from the cell itself. Soon, a hypothesis emerged that chloroplasts are symbionts, cells of photosynthetic bacteria that were once engulfed by a host and remained there permanently. Later, the endosymbiosis hypothesis was expanded to include mitochondria, organelles that consume oxygen and supply energy to all our cells. By now, this hypothesis has gained the status of a fully established, repeatedly proven theory. It is enough to say that mitochondria and plastids were found to have their own genome, mechanisms of division that are more or less independent of the cell, and their own protein synthesis system. Other endosymbionts have also been found in nature, which do not have billions of years of coevolution behind them and are at a shallower level of integration within the cell. For example, some amoebas do not have their own mitochondria, but instead have bacteria inside them that perform the same function. There are also hypotheses about the endosymbiotic origin of other organelles, including flagella, cilia, and even the cell nucleus. We can look at all these versions in more detail in one of the upcoming episodes. Would you be interested in that? Let me know with a like or comment. And turn on notifications so you don't miss anything. That will be all for today. Thank you for rating the video and for the opinions of advanced evolutionary biologists and experts in the comments below the episode. See you soon!